Welcome to Science Hangout from Science Mission. Today we have Dr. Charles Brenner, Chair and Professor of Biochemistry, University of Iowa with us. Dr. Brenner is an accomplished researcher who has published more than 100 research articles and won several awards. Dr. Brenner in 2016 won ASBMB Award for exemplary contribution to education. In 2015, he won keynote lecturer at the Brown University Pathobiology Retreat. In 2015, he was the Ackerson student lecturer. He gave the Ackerson student lecturer in biochemistry and redox biology at the University of Nebraska. In 2014, Dr. Brenner won the State of Iowa Regents Award for Faculty Excellence. So there are many more awards which he won throughout his career. So let's welcome Dr. Brenner. Dr. Brenner, welcome to Science Hangout.
Thanks, it's great to be with you.
So let me first congratulate you on your Nature Communications paper, which got published this morning.
Yes, thank you.
And let's get going. So can you please talk about your background, education, etc.?
All right. Well, I like to say I'm a country boy with a kind of a fancy education. I'm from outside of Boston, raised in New England, and graduate of New Milford High School in New Milford, Connecticut. Stayed in state to go to Wesleyan University.
Okay.
Graduated with a degree in biology in 1983. That was around the time that the biotechnology industry was getting going around Boston, around the San Francisco Bay Area. At that time, it was called the genetic engineering industry rather than biotech. And I went out to the San Francisco Bay Area, and I worked there for about 5 years in two biotech environments.
Okay.
One was Chiron Corporation, and the other was DNAX Research Institute in Palo Alto, California.
Okay.
And I trained in yeast molecular biology and yeast genetics in industry. And then at that point, I knew how to do experiments, and I wanted to know how to run a research group. And so I went off to graduate school at Stanford, and so stayed in the San Francisco Bay Area 10 years.
Okay.
Between my biotech experience and my grad school. Stanford, I worked with Bob Fuller in the biochemistry department, and I purified an enzyme called KEX2 protease, which is a pro-hormone processing enzyme. And great experience learning to do enzymology with Bob and in the biochemistry department at Stanford. And then 1993, moved back to the East Coast, and I trained in X-ray crystallography with Greg Petzko and Dagmar Ringa at Brandeis. And 1996, I got my first faculty position.
Okay.
And so I had a faculty position at Jefferson in Philadelphia, Thomas Jefferson University, from 1996 to 2003, and was recruited to Dartmouth in 2003, where I was on the faculty of the Norris Cotton Cancer Center, as well as the Departments of Genetics and Biochemistry at Dartmouth. And I expanded my program into NAD Metabolism at Dartmouth.
Okay.
And then 2009, I moved here to University of Iowa.
Okay.
And so it's been a great experience working in a number of really interesting environments.
Okay. And talk us about your mentors or role models who helped you throughout your career.
They've all been really, really important to me. So I was an undergraduate researcher at Wesleyan with a guy named Barry Kiefer, who was a Drosophila geneticist. When I was in industry, I had great supervisors. Tony Brake, who was a product of UC Berkeley. He worked with Jeremy Thorner at Berkeley. And then at DNAX Research Institute, I worked with Kuni Matsumoto, who's a very accomplished yeast geneticist. He's now a C. elegans geneticist in Nagoya in Japan. Bob Fuller, I mentioned, my grad advisor, is one of the dearest people in the world, and a really smart guy, a wonderful role model for how to ask the right questions and do thoughtful experiments. And then I had the great pleasure and opportunity to work for Greg Petsko and Dagmar Ringa when I was a postdoc at Brandeis, and I remain close to all of those, almost all of those people. Barry Kiefer has passed away, and I don't get to talk to Kuni Matsumoto very often, but Bob Fuller and Greg Petsko continue to advise me, and I try to be available to my former grad students and postdocs in the same way.
Okay. And I know you are a busy person with all the departmental work and research and all those things. So talk to us about something about your other than research hobbies or interests.
Well, that's a good question, right? So work-life balance.
Yeah.
So balance is really important, and so is the balance between energy intake and energy expenditure. So I like to exercise.
Okay.
One of my new hobbies is yoga.
Wow. Okay.
Doing hot yoga and doing vinyasa at 40 degrees C or something like that. And it's great to learn that. I kind of like getting into new things where I'm a rank beginner. I may not know as much as other people in a particular new area, but I like the sensation of noticeably improving from one day to the next. And so I very much enjoy things like riding a bicycle, doing aerobic physical activities, hiking, and the new hobby is yoga. I also enjoy gardening, cooking, following.
You do find time to do all those things.
I try.
Okay. Okay. So now getting back to the lab, like talk to us about your lab people, grad student, postdocs. So how do you manage them?
Yeah.
What is that experience?
Well, today is an exciting day because it's, as you said, the Nature Communications paper just came out. When it goes up on the lab website, it'll be called Trammell16c.pdf, meaning it's Sam Trammell's third first author paper of 2016. So Sam Trammell was our lab's most recent PhD. He's off in Copenhagen doing a postdoc. Wonderful person, trained in quantitative mass spec, and actually greatly improved our methods for targeted NAD metabolomics. And he was involved in everything from technology development to our first published animal experiments, which came out earlier this year, in which we showed that nicotinamide riboside is active in preventing diabetic complications, to our first human clinical trial, which came out today. We have more postdocs in the lab at any given time, usually, than grad students. Right now, I have Po Hyun Ir, who's a postdoc, Fan Bai, who's a postdoctoral research scientist, and Mark Schmidt, who's also a postdoctoral research scientist. Mark is an expert in targeted metabolomics, and he actually, right now, he's mostly doing targeted NAD metabolomics. Fan Bai is a mitochondrial biochemist, and Po Hyun Ir is broadly interested in metabolism, weight management, physiology of boosting NAD metabolism. Got a great technician in the lab, and Kita Chada, who supports all of our animal experiments. And for the last year, my lab has been having joint group meetings with two other principal investigators, Marie Migaud from Queen's University Belfast, who's been on sabbatical with me. And she's actually moving on November 1st to University of South Alabama, where she obtained a position as a full professor. And Amy Sindler. Amy's an assistant professor in health and human physiology here at the University of Iowa, but her lab space is located essentially embedded in the biochemistry department here at the university. And so the three of our research groups meet weekly and plan a lot of experiments together so that we have the power of organic chemistry and chemical biology from the Migaud group, and Amy's interest in vascular physiology, and then my group's support, which is in NAD metabolomics, all aspects of boosting NAD metabolism, and really anything that relates to NAD. We also have a project in our group that relates to epigenetic control of gene expression, in which we're really interested in how how RAS transformation and other oncogene changes lead to silencing of tumor suppressor genes. And I have a postdoc named Shilpa Thakur, who is really the one person in our group that is working on how DNMT1 and TET1 are dysregulated by RAS transformation.
Okay. And I know you have published so many good papers and all, but when you do the experiments, there is ups and downs and all. How do you motivate these kids when the experiments don't work?
Right. Well, you've got to have the right disposition to do science. So you've got to be patient, and you've got to be analytical, self-critical, aware of the literature, and you've got to communicate. So the people in my lab that do the best, example, Peter Belenke, who's an assistant professor at Brown University. Peter, in his about 5 years training as a grad student at Dartmouth, was more often in my office 2 times a day than 0 times per day. So, grad students are adults, and, some want to ask questions all the time, and some sort of don't want to ask as many questions because I don't know, maybe they're afraid of asking what they perceive as a dumb question, as some sign of something that they don't yet understand. But Peter and I just had a great working relationship, and he would pop into my office at any time to throw an idea out or ask whether I knew something about any new experiment that he had in mind. And it's really through that dialogue that we had a great working relationship and he had a high batting average of experiments that worked, I think in large part because we collaborated so well at designing experiments. And so I think that people who communicate well tend to do the best experiments. And so group meeting is really important, and open sharing of ideas is really important, and interdisciplinarity is important. And that's kind of how my lab is benefiting right now by having chemical biology, physiology, and biochemistry, metabolomics in the same group.
Okay. And you told us about NR, which you started working about 6 years ago or so. How did it come about? You had a kind of a shift in working.
Yeah. Well, longer ago than that, we published our first paper on the discovery of the eukaryotic nicotinamide riboside kinase pathway and cloning human nicotinamide riboside kinase 1 and 2. That was 2004. So we were at Dartmouth. It was shortly after we moved from Thomas Jefferson University to Dartmouth. So when I started my lab in Philly about 20 years ago, we really had a molecular oncology focus, and we were very interested in how tumor suppressor genes work. And I won't go into all of the twists and turns, but we ran into an enzyme called glutamine-dependent NAD synthetase. Yeast QNS1, human NAD-SYN1. And I was just quite interested as a biochemist in how that enzyme works. It has two active sites that communicate with each other. But in early 2000s, there was a rekindling of interest in NAD metabolism, because NAD went from being a redox cofactor, which is incredibly important, to not only a redox cofactor, but a consumed substrate of a class of enzymes called sirtuins, and also poly-ADP ribose polymerases, PARPs, cyclic ADP ribose synthetases. A number of enzymes use NAD, but not as a redox cofactor. They use NAD as a consumed substrate. And here I was, having cloned the gene for an NAD biosynthetic enzyme, I was able to address whether we understood the full range of ways in which a cell can resynthesize NAD from component parts. And it was in the course of doing those experiments that we discovered the nicotinamide riboside kinase pathway to NAD. So we discovered NR, nicotinamide riboside, as an additional NAD precursor vitamin. This was published in Cell in 2004 because it was rather surprising to discover a new vitamin kind of so late in the game. The NAD coenzyme had basically been a thing in biochemistry since around 1905, when Arthur Hardin called it cozymase as a fraction, a non-protein fraction that promoted glycolysis or cell-free fermentation. And in 1938, Conrad Elvehjem discovered nicotinic acid and nicotinamide, niacin, as vitamin B3, the commonly known precursors of NAD. So I was rather surprised that there was still another vitamin precursor of NAD that was out there, and that it took us until 2004 to uncover. And in the past, 12 years now, we've published almost 40 papers on NAD metabolism up until today, which is the first clinical trial of nicotinamide riboside. So it's gone from being literally something that was a plate assay in which we detected nicotinamide riboside in milk and identified the human nicotinamide riboside kinase pathway, in part by doing yeast. So literally we saw evidence of nicotinamide riboside on a petri dish. Today, we report human clinical activity of nicotinamide riboside. But the funny thing is that as an over-the-counter supplement, NR is currently being used by somewhere between 175,000 and 200,000 people a day, probably almost entirely in the US. So it's being used by almost 1 out of 1,000 Americans today as an over-the-counter supplement. 12 years ago, it was just a petri dish assay.
Okay.
Pretty amazing trajectory.
Yeah, let's look at your some of the work we have done in published in Nature Communications. If you could share the presentation.
Sure. Do you want some background?
Yeah, certainly. Go ahead.
Okay, yeah. Great. So I'll share presentation with you. Let's see if this shares with everyone. Can you see that?
Yep.
Fantastic. So now the first thing I have to do, is I have to give a disclosure because I have some, advisory activities with industry. So Chromadex, and can you see the pointer as well?
Yep.
Okay, great. So I'm on the scientific advisory board of Chromadex. This is a company that has commercialized NR. I'm a co-founder and chief science advisor of a company called Pro Healthspan that actually sells NR to the public. It's available at prohealthspan.com. And then I have my own little business that is aiming to do targeted metabolomics in order to kind of figure out who needs NR supplements and the effect of NR supplements on people.
Okay.
So just to kind of give people an orientation on classically what NAD is, this is NAD here on the left, and this is NADH here on the right. And so NAD is a redox cofactor in biology. And so when we're consuming macronutrients, I mean carbs, fats, protein, we're oxidizing fuels, and NAD becomes reduced to NADH, right? And then NADH becomes reoxidized to NAD plus at the mitochondrial inner membrane to make ATP. And then there are other processes in which NADH is reoxidized to NAD plus, like gluconeogenesis and forming ketone bodies. And you might also know that there are related compounds, NADP and NADPH, which has the phosphate here and the hydride here, that are important in other classical aspects of biochemistry. So NADP for making phospholipase and RNA DNA precursors, NADPH for detoxifying free radical species, making steroid hormones like testosterone and estrogen. And then right around the year 2000, Lenny Guarente and several others showed that a class of enzymes called Certuins have an NAD requirement as well. So now I'm depicting, if you look at the first way I depicted NAD, there's a nicotinamide moiety, and then there's the rest of this molecule. This is ADP ribose. So now I'm showing you ADP ribose linked to nicotinamide, and then an enzyme called a Certuin. Let me see if I can do a close up a little bit here. Range view.
Zoom it.
Zoom window. Okay, great.
Yeah.
So an enzyme called Certu or a Certuin will take a peptide with an acetylated lysine, like an acetylated histone, classically, and it will deacetylate that lysine so that you have a non-modified peptide. In the course of doing that, it uses NAD and it breaks the NAD into two pieces, a nicotinamide moiety and an acetylated ADP ribose.
Okay.
So it actually breaks NAD. And people were interested in SIR2 because SIR2 is a gene expression regulator in yeast. SIR2-related enzymes regulate a lot of protein functions in mammalian cells and in humans. And in particular, one of the important functions of SIR2 in yeast is that it's required for the longevity benefit of calorie restriction. So calorie restriction can extend lifespan in yeast and other model organisms. And so there are genes that respond to the level of glucose and the level of other macronutrients. SIR2 is required for the ability of low glucose to extend lifespan. And SIR2 requires NAD. And so a lot of people thought, gee, there must be some aspect of NAD metabolism that is affected by the level of glucose. And SIR2 is a key regulator of that. And so people got interested in how NAD is made. Incidentally, sirtuins, which are, and by the way, you can read more about this in this Tibbs review. Sirtuins do this reaction that I just depicted, and acetylated lysine becomes deacetylated, and that turns over NAD, and then PARP turns over NAD in polymerizing ADP ribose, and cyclic ADP ribose synthetases cyclize the ADP ribosyl moiety of NAD. So all of those things consume NAD. So, and by the way, let me point out that when you have DNA damage, poly-ADP ribose polymerase gets activated and degrades NAD in generating ADP ribose polymers, and that's involved in single-strand DNA repair. And if you take a look at this picture of an individual from 100 years ago that had a condition called pellagra, you notice that her sun-exposed skin has this dermatitis lesion, right? And so classically, there was a disease that we now understand was an NAD precursor deficiency, cured by improved diet and the discovery of nicotinic acid and nicotinamide as pellagra preventative compounds. Her sun-exposed skin is damaged in part because sun is activating PARP, which is degrading NAD, and so there's not enough NAD to do all of the vital functions in cellular metabolism that NAD is involved in. So people through much of the 20th century thought that NAD is kind of a solved problem because in 1938, LVM discovered these NAD precursor vitamins, and NAD was involved in this redox biology. And so people thought it's not so interesting any longer. And then all of a sudden, in the year 2000, Lenny Guarente and others, point out that there are very important non-redox roles of NAD. NAD gets turned over by enzymes like Certuin that are really important. PARP is really important. Certuin is really important. So people thought, gee, let's figure out how NAD is made, because probably there are very important regulators of NAD metabolism that explain things like longevity. Okay. So I wonder if anybody out in Google Hangouts land has ever watched a game of T-ball. Have you ever watched T-ball?
Yep.
You watched T-ball?
Yeah,
I
have some.
Okay, so this is the center fielder, okay, in a T-ball game. Okay? There's not a lot of action in the outfield, right? Most of the action is in the infield because the ball doesn't usually get out to the outfield. And when the ball finally goes out to the outfield, people get really, really excited, and everybody, the left fielder and the right fielder are coming in and trying to get this ball. This is me playing center field, okay? And I won't say who was playing left field and right field.
Okay.
But the ball came right to me. Okay. Because I was working on this enzyme, glutamine-dependent NAD synthetase, and people looked at this chart. This was published by 5 different research groups. It showed everything that was known about NAD synthesis. It has tryptophan coming in from the left, making NAD. There are 5 enzymatic steps here, 2 more enzymatic steps, and then NAD is made. My favorite enzyme is here. And then Certuin turns over NAD to nicotinamide, and then nicotinamide is converted to nicotinic acid. Nicotinic acid goes back up here. So according to this diagram, all roads to NAD went through my favorite enzyme. Okay? And when, in the year 2000, it appeared that NAD metabolism, or NAD metabolites, regulated Certuin, Dr. Guarente suggested that NADH would be present at high concentrations at high glucose, and that NADH inhibits SIR2. And that what calorie restriction would do is calorie restriction would lower the level of NADH.
Yep.
That would relieve SIR2 inhibition, and then SIR2 would work faster and the cell would have a more youthful gene expression program. So that turns out that's not really true. And then an outstanding scientist at Harvard, David Sinclair, suggested that the SIR2 product, nicotinamide, at its normal cellular concentrations, inhibited SIR2, and that a rise in an enzyme called PNC1, which converts nicotinamide to nicotinic acid, that's the key to calorie restriction. So it is true that high dose nicotinamide inhibits SIR2, but the level of nicotinamide in the yeast cell doesn't actually inhibit SIR2.
Okay.
And it's not really actionable either. So what we were working on was really just how we were not trying to explain longevity in 2004. We were just trying to understand how this enzyme worked. But what we discovered is that those steps that were in black were not the only steps in NAD metabolism. And we discovered, and I won't go through all of the many papers, but we discovered in blue the nicotinamide riboside kinase pathway. This is Pavel Bieganovsky, who was the postdoc in my group that found NRK pathway. And this is interesting because nicotinamide riboside kinase 2 is actually induced by DNA damage, or not by DNA damage, but by various types of damage, like in a damaged neuron or a damaged muscle. This pathway is induced. Shin Imai and others have shown that nicotinamide to NMN salvage is very important in vertebrate cells. This pathway actually declines in aging. We discovered NR as a metabolite and as a vitamin. We discovered nicotinic acid riboside as a metabolite, and all of these additional genes and enzymes in red, green, blue, a transporter in yeast. And you can see some of the trainees that did the work. This is Katrina Bogan, who got her PhD with me. And this is Peter Belenke, who got his PhD with me. And so we discovered, really a whole bunch of new genes and enzymes. And now nicotinamide riboside is probably considered the most promising and exciting compound called a STAC, or Certuin activating compound. And what you may know is that resveratrol was once proposed as a Certuin activating compound. This is a component of red wine. Resveratrol at very high doses does some very interesting things to mice, but it has a very complicated mechanism of action. Resveratrol does a lot of things, including inhibit cyclic AMP phosphodiesterases, and it really does not have good oral bioavailability. And so, in terms of the pharmaceutical industry, it's really been a huge financial consumer. And we think that nicotinamide riboside is really a very exciting compound that simply elevating NAD metabolism, it increases Certuin activity. So this is nicotinamide riboside, and our first publication in which we found it in milk. And unlike the discovery of nicotinic acid and nicotinamide, which were assays of kind of malnourished dogs, we didn't harm any animals in the course of discovering NR. And then in 3 years later, we showed the Certuin activating activity of nicotinamide riboside. So
Mm-hmm.
Here we're extending lifespan from about 12 and a half generations to 23 generations.
Mm-hmm.
You abolish that activity if you knock out the genes that convert NR into NAD.
Mm-hmm.
And this publication showed that increasing NAD metabolism with NR increases Certuin function in a yeast cell. And so NR is kind of special because it has a unique biosynthetic pathway. It doesn't cause flushing like nicotinic acid does. It doesn't inhibit sirtuins like nicotinamide does. In our newest paper, and I'll kind of skip to that, we show that it is a favored NAD precursor vitamin in the liver. You get more NAD from NR than you do from nicotinic acid or nicotinamide dose per molar dose.
Mm-hmm.
And it's also important because NAD declines in aging, and the ability to salvage one of the endogenous precursors, nicotinamide, also is reported to decline in aging. And so part of the interest in supplementing with NR that we do with NR supplements, such as those sold by Pro HealthSpan, some of the interest in that is to maintain youthful and optimal NAD metabolism as we age. So I think I will kind of skip forward to show you some of the things NR does in a metabolically stressed animal. So these are mice that are on a high-fat diet. They're on normal chow, you section their livers, they don't have a fatty liver. If they're on a high-fat diet, these white blobs are lipid droplets.
Mm-hmm.
They get a really high fatty liver. If you treat these mice with NR, there is much smaller lipid droplets. And these mice are actually type 2 diabetic on a high-fat diet. And with NR, they have much smaller lipid droplets, as you can see quantified. They have better liver function. They have greatly improved non-fasting glucose, much better glycemic control. And then these data, which were published earlier this year in scientific reports by Sam Trammell and other folks in my lab, show that with obesity and type 2 diabetes, in mouse, there's terrible diabetic complications. So the behavior of these mice shows that their nerves are shot, and microscopy shows their nerves are shot, and microscopy and behavior shows that their nerves are protected by NR. And we're going to be doing a clinical trial with Michael Abramoff here at the Iowa City VA. And then with Donna Hammond, we show in female rats that we can protect female rats from chemotherapy-induced peripheral neuropathy, and we can reverse that. And there's a clinical trial plan there. So now here's the thing, Sadashiva. I think I told you that 12 years ago, NR was just a plate assay, does all these amazing things in mice and rats. But I think I already told you 175,000 or more people are already taking it because it's an over-the-counter supplement. So we felt like the scientific that we're kind of getting out over our skis, right? Because as a natural product, companies like Chromadex were able to get GRAS status of it through the FDA and develop GMP manufacturing procedures for NR and simply go out there and sell it. Right? And so, what's the evidence that it's actually orally available and safe in a human being? So somebody had to start and take it. And so here's me. I was doing a little home construction renovation project at my house. And so somebody had to basically, be the first guy or gal to take it. And so I started with an N of 1 pilot experiment in which I informed the IRB at my institution that I wanted to take it, and I wrote out a consent document in which I said that I understood that there were probably not very many risks, but there might be a flush reaction or who knows what. And I was informed by the IRB that if I took it myself, it was not considered human subjects research.
Okay.
There's a long history of N of 1 trials in which people take their own stuff, but I was expressly forbidden to give it to anyone else. I'm a department chair, so I certainly couldn't give it to anybody in my lab or anybody in my department, but I could take it myself.
Okay.
So I started off doing a pilot experiment where I took a gram a day for 7 days. And sure enough, my NAD went up in my blood. So essentially nothing happened for the first 4 hours. And then at just after 4 hours, my NAD went up by about 2.7-fold. Surprise, another metabolite called NAD went up even more, 45-fold. This was unexpected. So we went into mice. I won't go into all of the data. It gets very complicated. But we were able to compare NR, nicotinic acid, and nicotinamide in a very defined time course in mice. And these data showed that NR boosts liver NAD metabolism in a mouse to a greater degree, this is in black, to a greater degree than nicotinic acid, which is in blue, or nicotinamide, which is in green, and with a different time course and through unique NAD metabolites. And that this NAD metabolite comes up in mice just as it does in humans. These data also showed that that NR boosts Certuin activity to a much greater degree than nicotinamide and to a higher degree than nicotinic acid. And then we finally, with the N of 1 experiment where I had shown in my own blood that my NAD metabolism was boosted, and in the mouse in which we found this biomarker and we kind of confirmed the time course and the dosing of nicotinamide riboside, we went to a properly consented IRB approved human clinical trial with 6 men and 6 women in which we saw this nice dose-dependent increase in NAD metabolism, multiple metabolites. And we showed with 3 different doses of NR, 100 mgs, 300 mgs, and 1 gram, that single doses of NR give an elevated NAD in the blood 24 hours after taking it. And so this is the citation of what came out today. So we've learned, a whole lot in 12 years that it's a unique precursor, and I won't tell you everything that we've learned about it, but it's in part it's valuable because of the NRK pathway and the properties of NRK1 and NRK2. And we learned in this study that we get more NAD with NR than with the other precursors. There's no flushing. There were no serious adverse events reported. There's a tremendous research activity with a number of diseases and conditions in mice and rats, and we're testing those things in humans. And so we're really excited about NR research. And of course, I couldn't have done it by myself. And so these are the people in my lab and the people with whom that I've collaborated, and the folks that have given us funding. And again, I'm happy to take questions from you or viewers about our research.
Okay, so let me see if anybody has any questions. Meanwhile, I was thinking this NAD, that metabolite, right? Does it get excreted in the urine or it is just in the blood?
So NAD doesn't so much get excreted. The stuff that gets excreted into the urine doesn't have a phosphate on it.
Oh, okay.
So what you largely see in the urine are methyl 2-PY, methyl 4-PY, methylated nicotinamide. And so so NR boosts. The first thing that happens is it goes into the cell, gets converted into NMN by nicotinamide riboside kinase, then to NAD, and then to NADP, right? And NADP, NADPH, NADH. The interesting biomarker metabolite, NAD gets deamidated to NAAD. That stays inside the cell, and it can get converted back to NAD+. Once NAD gets converted to nicotinamide by, for example, a Certuin, then the nicotinamide can either be salvaged back up to NAD, or it can be methylated and excreted.
Okay. And what is the half-life in the body, basically? Like, you need to take it every day. How does it go?
So most people that take NR supplements, True Niagen, for example, take it in the morning. 225 milligram capsules is quite a lot more than in a glass of milk. It's hundreds of times more or thousands of times more than in a glass of milk. And of course, it doesn't have any calories because it doesn't contain the macronutrients that the calorie-containing macronutrients. And we know from doing the clinical work that our NAD metabolism is still higher 24 hours after taking an NR supplement. The peak NAD is probably about 8 hours after taking NR. Some people I know take NR twice a day and have reported improved sleep by taking NR both in the morning and the evening. The sleep has not been clinically proven. There's a number of things that have been observed. I would say it's anecdotal, but it's widely observed. People report a greater feeling of vitality, quote, more energy, more youthful appearance, athletic performance, improved skin. Some people say that their hair and fingernails grow more rapidly. I've seen improved digestive function, which is interesting because pellagra has a strong diarrhea component, was observed as a condition of skin dementia as well as diarrhea in the olden days when there were populations of people that frankly didn't have enough NAD precursors. I have enough NAD precursors in my diet to avoid pellagra without taking NR, but when I started taking NR on a daily basis, I observed improved digestion. That's the kind of thing that's going to be clinically tested as well.
Okay. And you mentioned that NAD goes down with the age, right?
That's been widely reported. So it was first reported in mice, and there are some additional reports of NAD declining in human beings as well, yes.
So does it help, like we take some of these things after 50 and all those, like some of the vitamins we take after 50, which is slightly different than what you don't take and all. So would it be helpful, like for the people who are older, this having So
the idea is that we know that our metabolism slows in aging. We know that mental function declines in aging, that muscular function declines in aging, and interestingly, NAD is central to all of those processes. NAD declines in aging, and so it's thought that by boosting NAD metabolism with a non-flushing, non-Certuin inhibiting NAD precursor vitamin, and NR is really the only thing that we know of that will increase NAD without causing flushing and without inhibiting sirtuins. We think that by boosting NAD metabolism, that we can potentially age better. And so that's absolutely the idea. Of course, at this time, NR is being marketed as an over-the-counter supplement, so it's not intended to treat any disease or condition. And your mileage may vary, but it's absolutely something that can be tried. And some people keep a journal, and but it's going to be very important as we do more clinical testing that's placebo-controlled, in order to really test conclusively whether NR has some of the activities in a human being that it has in a mouse and a rat. Because frankly, in a mouse and a rat, the more metabolically dysregulated the mouse is, the worse the diet that the mouse has, and the older the mouse is, the more profound the effects of NR has. So in a healthy mouse, it's hard to see the effect of NR. In an overfed mouse, or a mouse with hearing loss, or a rat that's been treated with chemotherapy, the effects of nicotinamide riboside are profound.
Yeah.
I want to say one other thing, if I may, about clinical testing.
Yeah.
So there are some nutritional supplements like selenium that have profound effects, but have been difficult to detect the beneficial effects of selenium, even though they would have been anticipated from preclinical work. And the thinking is that when selenium got to clinical testing, the people that were interested in participating may have wanted selenium.
Yep.
Right? So they may have gone to GNC or Whole Foods or whatever and gotten selenium. And so effectively everyone on trial, the experimental group as well as the supposed placebo group, may have been taking selenium.
Yep.
I've got a solution to this problem, the biomarker. NAD itself is a pretty abundant metabolite. It's just like testosterone. Every male has a level of testosterone. You can tell who's juicing, right? Who's taking a testosterone supplement when you do targeted mass spec on the androgen metabolome, and you look for low abundance metabolites, right?
Yep.
That way they can see the specific metabolite that a person might be taking, and other metabolites that are normally at a low level, that get to be at a much higher level when a person takes those kinds of supplements. Well, we discovered that NAAD is normally below the detection limit in a person that's not supplementing, and it goes up to almost 1 micromolar in a person that has taken NR. So by using the information that we just published today in Nature Communications, we'll be able to monitor clinical testing and be able to observe not only increases in NAD metabolism, but we'll actually be able to exclude any participants that are supposed to be in the placebo group that are actually cheating and are taking NR.
Yeah. And do you have any ELISA test or anything for NAD?
ELISA kind of things. So that's a great question. So the way that we measure the NAD metabolome is by LCMS, right? Yeah. Which means that you almost have to be in my lab. Yep. You've got to have a lot of specialized equipment, extraction and everything. And so you're pointing out, do we have a cheap, inexpensive way, like a kit, for detecting NAD? And that's actually part of the reason that I established the company NRomics, to develop those kinds of kits and tools that we could commercialize and disseminate that would allow people to measure increases in NAD metabolism, as well as decrements and deficiencies in NAD metabolism without the complicated machinery and facilities that you require for mass spec. So short answer is no, I don't have a kit for NAD or for boosted or for decremented NAD metabolism, but we're very much interested in developing those things.
And the other question is, like you mentioned about sleep and all. So is there any work planned regarding circadian rhythm?
Yeah. So it's a great question. So NAD metabolism, like many other things, is highly regulated by circadian cycles. And so in the mouse, NAD metabolism peaks at night, which is when mice are active and they're eating.
Mm-hmm.
That's why people that are supplementing supplement when they get up, right?
Mm-hmm.
So this is another important point, is that although there's NR in milk,
Mm-hmm.
the real most important sources of NAD metabolism are actually in food. I like to say whole foods with a small w and a small f. So when we eat cellular stuff, whether it's meat, vegetables, grains, we're breaking down cells. And when we break down cells, there are abundant metabolites like NAD, NADPH, that get broken down into non-phosphorylated compounds, including NR,
Mm-hmm.
nicotinamide, nicotinic acid, that then get taken up by cells and get converted back into NAD. So while milk is kind of unique in that its NAD metabolome does not include any cellular metabolites, because it's not a cellular material, it's an extracellular material. So the NAD metabolome in milk consists of NR and nicotinamide. We published that in the Journal of Nutrition earlier this year. That was actually Trammell 16A 2016.
Wow.
The diabetic neuropathy study was Trammell 16B
Mm-hmm.
PDF. And today's paper, Trammell 16C. But milk is unique in that it has NR. Other foods contain, essentially get broken down. But we don't have control over the degree to which food gets broken down into NR or nicotinamide or nicotinic acid, which are sort of less valuable NAD precursors. And that's why supplementation makes some sense, especially as we age.
Okay, and the last question is regarding chemotherapy.
Yes.
So you mentioned like there is a paper published. It helps with the people with the chemotherapy.
Not yet published. So
Not yet. Okay.
There's work that has been presented at meetings and
Okay.
In abstracts and in grant proposals, in which, and this is a collaboration with Donna Hammond, Marta Hamady, PhD, in Professor Donna Hammond's lab, in which we've tested rats for their resistance to chemotherapeutic neuropathy, and nicotinamide riboside has strong activity in that assay. And on the basis of the very exciting preclinical data that the Hammond group and our group demonstrated, we have been funded to do a phase 2 clinical study of women undergoing cancer chemotherapy with paclitaxel to see whether NR will allow them to complete chemotherapy on paclitaxel. So we're excited by the possibility that NR prevents chemotherapeutic neuropathy in human beings, but that's not yet been demonstrated.
Oh,
it's been demonstrated in a rat.
Rat, okay. Yeah, that would be interesting because I have heard a lot of people having that neuropathic problem with the chemotherapy. So if it works out, that would be a very good discovery, and certainly you can add one more feather to your cap.
Well, you know, it's a great story, and it relates to the fact that NAD is degraded in the course of neurodegeneration, and part of our group is engaged in work to try to understand the mechanism by which NAD is degraded in neurodegeneration and the mechanism by which NR protects. You know, some people think that it relates entirely to Certuin activities. We think that Certuins are important, but we think that resistance to reactive oxygen species damage is also very important, and that by boosting the NAD metabolome, including NADPH, in liver, in a damaged neuron, and in many other tissues, that NR is protective.
Okay, I think we are almost out of time. It's been 1 hour. Thank you very much for taking your time, precious time, and come and talk to us. And I think the viewer must be really delighted. And we have, I think, I don't know if they are from your lab. There are a few viewers today, so which is very interesting.
Great.
Thank you so much. So I would like to again talk to you when your next Nature Communication paper comes out.
Great. Thanks, and thanks to all the people that watched. You can find our lab at Brenner. I think it's biochem.uiowa.edu/Brenner. Thanks.
And people can email you, right, if they are interested?
Yep. Okay. Absolutely.
Okay. Thanks a lot.
Take care. Bye.